Antibiotic susceptibility test. Importance of antimicrobial susceptibility testing is to know the susceptibility of organism. That means whether the organism is susceptible to particular drug. To check the potency of antimicrobial agent means to check the efficiency of antimicrobial agent, to check the concentration of antimicrobial agent, whether the concentration is appropriate for killing the organism, to detect possible drug resistance, whether the organism is resistant to particular drugs or antibiotics, and also for choosing appropriate drug for treatment. Antibiotic susceptibility test method is classified into diffusion test and dilution test. In diffusion test, we have Kirby-bauer disk diffusion method, Stokes disk diffusion method, E-test or Epsilometer test. In dilution test, we have Broth dilution method and Agar dilution method. In that, the diffusion test, the principle is based upon the diffusion of antibiotics to the medium. The diffusion means the movement of substance from high concentration to low concentration. In the dilution test is based upon the dilution of antibiotic. That means serial dilution of antibiotic is taken. To that we will add the test organism and MIC is calculated in the dilution test method. Kirby-bauer disk diffusion method. This disk diffusion method is a gold standard method we are using for confirmation or testing the susceptibility of bacteria. And this standard disk diffusion method was introduced by Bohr and Kirby in 1956. So this is called as disk diffusion method. Why it is called as disk diffusion method? From the disk, and this disk is coated with non-concentration of antibiotics, and from the disk the antibiotics will diffuse into the medium. You can say this is the antibiotic disk, and the antibiotic will diffuse in the medium and it will kill the bacteria which is near to the disk. So we will get a zone like this. So these are the clear zone, and by seeing this zone, we will get an idea whether the organism is susceptible or resistant to this particular drug. Mullahinda agar or MHA. This MHA or Mullahinda agar is used for this antibiotic susceptibility testing in the case of this diffusion method. And this Mullahinda agar is a non-selective or non-differential medium. Because of this non-selective and non-differential medium, most of the organism will grow well in this medium. And also additionally, this Muller-Hinder agar will contain starch. And the starch will absorb the toxin released by the bacterium so that the bacterial toxin will not interfere the antibiotics. For this disk diffusion method, we need a test organism, Mullahinda agar, cotton swab, 0.5 McFarland standard, antibiotic disk, and measuring scale. Preparation of inoculum from primary culture plate. So this is the primary culture plate. Here you can see there are so many colonies are there in the primary culture plate, and each colony represent single type of organism. So one colony we will take with the help of this inoculating loop, and we will transfer or we will inoculate this single colony into a liquid culture or broth, and this broth may be sterile water, nutrient broth, or peptone water, and this is the first step. And after that, the inoculum we will compare with this 0.5 McFarland standard. So this is our inoculum and this is the McFarland standard. And this McFarland standard is a reference standard to check the turbidity or to compare the turbidity of the inoculum. So that our inoculum will be standardized by comparing this McFarland standard with our inoculum. And after that, we will swap the inoculum into the culture medium. This is our inoculum. With the help of a sterile swab, we will take the, we will just dip the sterile swab into the inoculum and we will just lawn culture the swab to this Muller Hinder Agar. Lawn culture means uniformly we will spread the inoculum to this Muller Hinder Agar. Next step is the application of discs to the media. So this is the antibiotic disc. Antibiotic disc. And this is the antibiotic disc. And this antibiotic disc is a small circular 6 mm diameter filter paper soaked with antibiotic or impregnated with antibiotic. That means simply we can say that. And this 6 mm filter paper will contain antibiotic, and this we will used for antibiotic susceptibility testing. So in this application of disc to this media, we will take the disc. That means depending upon the organism, the selection of disc will be or antibiotics will be different. And this disc we already told that is a filter paper disc impregnated or coated or added with this antibiotic with non-concentration. Just we will place the antibiotics disc to this lawn cultured Muller-Hinder agar medium. Manually we can dispense or we have a dispensaries that is done pre-prepared dispensaries there. With the help of dispenser just we will place the antibiotics disc into Muller-Hinder agar. So after the disc suspension is over, so this is the disc suspended medium. We will incubate this medium at 37 degrees Celsius for 16 to 18 hours or overnight incubation. And after the overnight incubation, we will get an observation like this. So after incubation, this will be our observation. Here you can see some clear area or zone we can see here around the disc. So these are the clear area we can see here. And this clear area means the bacterial growth is not seen in this area, and all the other places or area the bacterial growth can be seen. So after the incubation, we will get an observation like this. And these are the antibiotic discs you can see here. And in some area, we can see that there is no zone. The bacteria is grown all over the area. And in some area, you can see the zone of inhibition. Here you can see these are the zone of inhibition. And this zone of inhibition we can detect by simply seeing the clear zone around the disc. And this is a disc diffusion method. In disc diffusion method, what will happen? We already told that this disc will contain antibiotics. If the bacteria is susceptible to this antibiotics, what will happen? From the disc, the antibiotics will diffuse into the medium. You can see the antibiotic will diffuse into the medium. Due to the diffusion of these antibiotics around the antibiotic disc, the bacterium will die. So here you can see this is the bigger zone of incubation. That means the diffusion is more here, and this bacteria is susceptible to this antibiotic disk. And here you can see there is no zone of inhibition. That means this bacteria is resistant to this particular drugs. Resistant means during incubation and this disk will contain antibiotic. This antibiotics will diffuse into the media, but this drugs will not kill the bacteria. That is why we will call it as resistant. Measuring of diameter of this zone of incubation. Here you can see with the help of scale, we will measure the zone of inhibition and we will note down that zone of inhibition. The measured zone of inhibition we will compare with this standard chart of interpretation. This is the standard chart. This is the antibiotic drugs. This is about the disk strength. This is the diameter of zone of inhibition, that is resistant, intermediate, or sensitive. For example, if you are taking the antibiotic as amikacin, that means we are testing the organism with amikacin, and if you are getting the zone of interpretation or the zone diameter is less than this 14, we will call it as that particular organism is resistant to amikacin. And if we are getting the zone measurement is 15 to 16, in between 15 to 16, we will call it as that particular organism is intermediate to amikacin. If you are getting the zone of diameter, zone diameter greater than 17, we will conclude that that particular organism is sensitive to this amic acid. That means whatever zone diameter we are getting, that we will compare with the standard chart of interpretation. And we will conclude and we will give the result as whether the organism is resistant, intermediate, or sensitive to that particular organism, to the particular drugs. Interpretation of sound. The sound can be interpreted whether it's sensitive, moderate or intermediate, or resistant. The sensitive means the diameter will be equal or greater than the standard. The diameter what we measured will be equal or greater than standard, or we will call it as sensitive. And the diameter is moderate or intermediate, we can say that the result is, or our interpretation is intermediate. The resistance means there is no zone of inhibition or less than the moderate, we will call it as resistant. In Kirby-bauer disk diffusion method, first we will take the Muller-Hindert-Agar plate. To this Muller-Hindert-Agar plate, we will lawn culture the test organism, and after lawn culture the test organism, we will apply or we will add the antibiotic disc. Depending upon the organism, the selection of the antibiotic disc will vary. And after adding this antibiotic disc to this Muller-Hindert agar, we will incubate the plate at 37 degrees Celsius for 16 to 18 hours or overnight incubation. And after that, we will get the result like this or observation like this. That means in some discs we can see the more zone of inhibition, or in some discs we can see there is no or less zone of inhibition, and we will measure the zone of inhibition by the help of scale. And after that, we will compare the zone of inhibition with the standard chart. And we will interpret the result whether the organism is resistant or sensitive or intermediate to that particular drug. Stock disk diffusion method. In this stock disk diffusion method, the principle is same as that of the Kirby-bauer disk diffusion method or diffusion method. That means the antibiotics is diffused into the medium. So the surrounding whatever organism is present, that organism is killed by the action of this antibiotic. Preparation of inoculum from primary culture plate. So this is the primary culture plate. Here you can see there are so many colonies are there in the primary culture plate, and each colony represents single type of organism. So one colony we will take with the help of this inoculating loop, and we will transfer or we will inoculate this single colony into a liquid culture or broth, and this broth may be sterile water, nutrient broth, or peptone water. And this is the first step. And after that, the inoculum we will compare with this 0.5 McFarland standard. So this is our inoculum, and this is the McFarland standard. And this McFarland standard is a reference standard to check the turbidity or to compare the turbidity of the inoculum, so that our inoculum will be standardized by comparing this McFarland standard with our inoculum. The shaping of inoculum to Muller agar after checking the McFarland standard, 0.5 McFarland standard, or comparing the McFarland standard, we will make three parts in the Muller agar in the case of stock culture method. First and the third part is control. Here you can see this picture, and the center part is called as test. And we will launch culture the control strain into the first and third part. The test organism is launch culture into the center part of the Muller-Hinder agar. After launch culture, we will incubate the culture plate at 37 degrees Celsius for 16 to 18 hours or overnight incubation, and later we'll observe the zone of inhibition. Here, this is the Muller-Hinder agar plate. This Muller-Hinder agar plate we will divide into three parts. The first part, second part, and the third part. This is the first part. We will inoculate the or we will launch culture the control strain, and this middle part, the second part, we will inoculate the test strain, and the third part, we will inoculate the control strain. That means the first and third part we have the control strain, and the center part we will launch culture the test strain. After that, we will keep the antibiotics this in between these two portions, like that. That means after incubation, we will get the result like this. And after incubation, we will compare the zone diameter of control to the test. Control to the test. While comparing the zone diameter, you will get an idea about whether organism is resistant or sensitive to particular drug. In stock diffusion method, we have one more method is there, that is modified stock disk diffusion method. Here we have only one difference. We already told that the Muller-Hinder agar we will divide into three parts: first part, second part, and third part. In stock disk diffusion method, the center portion we will launch culture the test strain, and all other portion, the first and third part, we will add the control strain. In this modified stock distribution method, the center portion we will launch culture the control strain, and the other portion, the first part and the third part, we will launch culture the test strain. After that, we will compare the control strain diffusion or the zone of inhibition with the test strain. Epsilometer test. And what is this epsilometer test or e-test? The e-test is used, or epsilometer test is used to determine the MIC, or by doing this with the help of this e-test or e-strip. This is the e-strip. We can calculate or we can predict the MIC of the organism. And you can say this is the strip, and this is a plastic strip having different concentration of antibiotics. So you can see this is 1, 1.5, 2, 3, 4, 6, 12, 24, 64. Like that, the concentration gradient is increasing. And what concentration? The antibiotic concentration gradient is increasing from one side to another. So by applying this antibiotic or this applying this Epsilometer antibiotics into this Muller Hinder agar, we can calculate or we can observe we can predict the MAC of the antibiotics towards the particular organisms. The E-test procedure, and this is the culture media containing culture or organism. From here, we will collect a single colony. Just we will, the help of inoculating loop, we will inoculate the single colony into this broth culture or liquid media, and after that we'll incubate. After incubation, we will compare this broth or liquid culture with 0.5 McFarland standard. And after attaining this 0.5 McFarland standard turbidity, we will inoculate or lawn culture the organism into Muller Hinder agar. And after lawn culture, just we'll apply the E-strip to the Muller Hinder agar. We can place single disc or multiple disc depending upon our requirement, and after that we will incubate the medium containing E-strip for overnight or 18 to 24 hours depending upon the organism. The incubation will be varies, and we will observe the zone of incubation, and by seeing the zone of incubation we can determine the MAC of the antibiotics towards the particular organisms. So after incubation we will have different observation depending upon the organism towards the E-strip of the antibiotic, because the different organism will have different observation depending upon the strip what we are using. So how we will calculate or how we will predict the MIC of this organism? The MIC means minimum inhibitory concentration. That means here you can see the minimum inhibitory concentration of these drugs for this bacteria is 0.25 milligram per ml. So this 0.25 milligram per ml is the minimum inhibitory concentration of this drug for this bacteria. Because we already told that this is a plastic strip coated with different gradient of or different concentration of antibiotics. Here you can see the antibiotic concentration is increasing from this side to this side. So here you can see 0.25, 0.38, 0.75, 1, 2, 3, like that, the concentration gradient is increasing. And the zone of inhibition is starting at this area or this point, that is 0.25 milligram per ml. So this 0.25 milligram per ml is the minimum inhibitory concentration. Tube dilution for antibiotic susceptibility testing. And this tube dilution is also called as broth dilution method because here we are using liquid media. And the graded amount of drug is taken into Muller Hinder broth. That means the graded or the concentration, different concentration of drug is taken in Muller Hinder broth, and this Muller Hinder media are inoculated with test bacterium, and this control strain is also inoculated in this broth, and we will incubate at 37 degrees Celsius for overnight incubation or 16 to 18 hours. At last, the end point is taken as MIC or MBC. MIC and MBC. MIC means minimum inhibitory concentration. That means the minimum amount of antimicrobial agent required to inhibit the growth of particular organism. That means minimum inhibitory concentration means that will inhibit the growth of that particular organism. In MBC means minimum bactericidal concentration. That means the minimum amount of antimicrobial agent or drug required to kill the bacteria. That we will call it as bactericidal concentration. Bactericidal concentration means
that will kill the bacteria, and inhibitory concentration means that will inhibit the particular growth of bacterium towards that particular.
Tube dilution method. The tube dilution method is also called as Broth dilution method. Here, the test organism is inoculated into tube containing serial dilution of antibiotics. That means first we will take the test tube. In this test tube, we will add equal volume of Muller-Hinder broth. To this Muller-Hinder broth, we will add different gradient of different concentration of antibiotics in serial dilution. Here you can say the concentration of antibiotic is 64, here 32, 16, 8, 4, 2, 1, like that. At last, this is the control. Like that, we are serially diluting the medium with antibiotics. And after that, we will incubate these tubes for overnight incubation or 60 to 18 hours at 37 degrees Celsius. And after that, we will read the result. And after the incubation, here you can see the first four tube doesn't show any visible turbidity. And from here onwards, we can see the turbidity in the culture broth or culture medium. This turbidity indicates the growth of the bacterium. And this endpoint, we will consider as the minimum inhibitory concentration, that is 8 milligram per ml. The minimum inhibitory concentration is also called as the amount of antimicrobial agent required to inhibit the growth of bacteria, that is called as MIC. And to calculate the MBC, that is minimum bactericidal concentration, again what we will do, we will subculture the broth into separate solid medium, because till here there is no visible turbidity is not seen in these tubes, and we have to confirm whether the organism is present in this tube. From here onwards, we know that the turbidity is present. The turbidity indicates the growth of the bacteria. And the first four tube doesn't show any visible turbidity, so that to confirm whether the growth is present or not, we will subculture the each tube into separate solid medium. And after that, we will observe the growth in the solid medium. And after the subculture into solid medium, we will incubate the solid medium at overnight incubation, that is 16 to 80 degrees, 16 to 18 hours for 37 degrees Celsius. And we will observe whether any growth is present or growth is observed in this solid medium. Here you can see the first two plate doesn't have any growth, and the third and fourth plate is having growth. So this endpoint, that is 32 milligram per ml concentration, we will consider as the MBC, that is minimum bactericidal concentration of this antibiotic. So this 32 milligram per ml is considered as the MBC. That means this 32 milligram per ml can kill the bacteria. Agar dilution methods. In agar dilution methods, serial dilution of drugs are prepared in Muller-Hinder agar and poured into plate into which the bacteria are inoculated. Advantages are it can determine the MIC, that is minimum inhibitory concentration of an antimicrobial agent against bacteria. Many strains can be inoculated on each plate containing antibiotic dilution. So, first step is the preparation of optimized media containing drug. So, to this Muller-Hinder agar media, we will add non-volume of antibiotic or drugs. The second, different concentration of the drugs are prepared by dilution with water. To this non-concentration of media containing antibiotics, we will mix with water so that we will get different concentration of antibiotics in different test tube here. And the media containing different dilution of drugs is poured in agar plate, which are then inoculated. That means the serial diluted media containing antibiotics we will pour into petri dish, and after pouring this media into petri dish, we will inoculate the bacteria into this petri dish, and we will incubate and observe the result. And this is the picture of serial dilution of drugs in Muller Hinder Agar medium. Here you can see the 64 microgram per milliliter antibiotic concentration is present in this Muller Hinder Agar. Here it is 32, 68, 4, 2, 1 microgram per milliliter drug concentration is present in this particular Muller Hinder Agar medium. And this one is the control, and this control will not have any antibiotics. Plain Muller Hinder Agar, we will keep it as control. And after that, to this medium, to each medium, we will inoculate the bacteria. After inoculation, we will incubate this each plate at 37 degrees Celsius for overnight incubation for 16 to 18 hours. So after incubation, we will get the result like this. You can see this three plate doesn't have any growth, and the growth is starting from this fourth plate, that is 8 microgram per milliliter. Later, the growth is increasing, and this is one is the control, and control is also having sufficient growth. So, if you are calculating the MIC, we can see that this 8 microgram per milliliter is the MIC of this particular drugs towards the bacteria. Another advantage of this agar dilution is many strain can be inoculated on each plate containing antibiotic dilution for knowing the MIC of the organism towards the antibiotic. Here the procedure is same as the earlier what we discussed. First we will make different concentration of antibiotics in Muller Hinder Agar. Here you can see this is 64 microgram per milliliter antibiotic concentration. Here the 32, here 16, and here 8 milligram per milliliter antibiotic containing Muller Hinder Agar medium. And this equipment is called as multiple inoculator or stir replicator. And we told that in this agar dilution, many strains or multiple strain can be inoculated on Muller-Hinder agar of different concentration of antibiotic for calculating the MIC. So this multiple inoculator or stirred replicator can inoculate multiple strains. Here you can say these are the wells, and this well will contain different type of, each well contain different type of strains or microorganism or bacteria, and this is the probe of this multiple inoculator. If you are dipping this probe into this well, the organism will be transferred. The organism can be inoculated into this Muller-Hinder agar at a time. That means at a multiple inoculation can be occur in one step method. So in this procedure, this is the Muller-Hinder agar containing different concentration of antibiotics. And with the help of this multiple inoculator, just we will inoculate the bacteria or different strains into this each Muller-Hinder agar having different concentration. And after that, we will incubate it for overnight or 60 to 18 hours. And after the incubation, we will get the observation like this depending upon the organism and the concentration of antibiotic what we used. So by seeing this growth in each plate, we can interpret the MIC, that is minimum inhibitory concentration of antibiotics for bacteria.
So the antibiotic susceptibility testing will help us to know the susceptibility of organism towards the drugs, and also we can check the potency of antimicrobial agent, and also we can predict the concentration of antimicrobial agent towards different organism, and also we can detect the possible drug resistance, and also this antibiotic susceptibility test will help in the treatment for choosing appropriate or proper or accurate drug for the treatment of the patient.
Subscribe to Simply Micromed. If you're curious about medical microbiology, infectious diseases, and unraveling the mysteries of tiny organisms, hit that subscribe button. Join our community of science enthusiasts, and let's explore the microscopic world together. Remember, knowledge is contagious. Spread it by liking and subscribing. Thank you.